Hello everyone, I'm Dr. Ramya. I'm an oral physician and a maxillofacial radiologist, and I'm a researcher too. And I'm here to present my work on development of 3D printed natural fiber reinforced composite scaffolds for maxillofacial reconstruction. These are the following contents which I'm going to talk in my coming slides. What exactly is a maxillofacial region? It includes the bones of the skull, frontal, parietal, occipital and temporal, and the other facial bones, which includes sphenoid, ethmoid, nasal bone, zygomatic bone, maxilla, and the mandible. A need for maxillofacial reconstruction arises due to congenital or acquired disorders like trauma, tumor, or infections. There are various treatment options which are available in the form of autografts, allografts, or xenografts. As we know that the gold standard treatment protocol is autografts, but still it comes hand in hand with that of various advantages and disadvantages, like requirement of a second surgical procedure, donor site morbidity, and it is expensive. So the next options will be in the form of allograft or xenografts, where the bone graft is derived from a same species or from a different species. But yet these two options also carry the risk of infections and the patient is subjected to long-term immunosuppression. And the third options are in the form of various natural or synthetic biomaterials like collagen, alginate, gelatin, various ceramics, tricalcium phosphate, bioglass, etc. But all these materials have poor mechanical strength and they're handicapped by their brittle nature. So the last options will be in the form of metallic implants, which includes titanium and stainless steel. These metallic implants have very high strength when compared to that of the native bone, thus leading to stress shielding effect, soft tissue inflammation and marginal bone loss. So, what should one consider when they're trying to develop a biomaterial for bone regeneration? These five important material characteristics for optimal bone repair and regeneration should be taken into consideration. The material should be biocompatible, bioactive, biodegradable, it should be mechanically stable, and it should be 3D printable. As we know that the strength has to balance with that of the native bone, because if the strength balances, then that leads to the better bone repair and regenerative procedures. When it comes to craniomaxillofacial reconstruction, aesthetics plays a very important role for social and psychological well-being of the patient. So 3D printing and customization plays a very important role here. Coming to the literature review, there are a lot of various 3D printed scaffolds where they are working on different biomaterials. So one such biomaterial is on 3D printed PCL scaffolds, where they have cultivated mesenchymal stem cells on these PCL scaffolds. And they have found that the control group, that the 2D plate, has more number of viable cells when compared to that of 3D printed PCL scaffolds. So another group where they have mixed PCL with beta tricalcium phosphate and have 3D printed it. They could see good bioactivity, but at the same time, because of the brittle nature of beta tricalcium phosphate, the composite has shown less strength when compared to that of pure PCL. So the scope of the research is to develop a novel natural fiber reinforced composite. So in our study, we have utilized silk fibroin microfibers and have reinforced these microfibers into the PCL matrix. And the justification why we have used these two input materials is PCL, which is polycaprolactone, synthetic polymer, it is biocompatible, low melting temperatures about 59 to 64 degrees centigrade so that it can be processed at low temperatures. It is 3D printable as it is thermoplastic, but the drawback here is it is hydrophobic and it lacks cell-specific recognition sites. So, to counteract this, we have added natural polymer, that is silk, which is also biocompatible and it blends with any other natural and synthetic polymers and it is already FDA approved biomaterial for medical applications. Another important thing, this silk fibroin possesses an extraordinary property to stimulate the bone repair and regeneration. What exactly is silk? Silk is present in the glands of silk producing anthropoids like silkworms, spiders, scorpions, etc. And this silk has two types of proteins, sericin and fibrin. Sericin is a coat which is a glue-like protein, accounts for about 30% of the whole content, and the fibroin accounts for about 70% of the whole silk protein. Two types of silk variants, Bombyx mori and Antheraea mylitta, are being considered in our study. The main reason for selecting these two silk variants is to compare, because this Antheraea mylitta, which is a wild variant, it consists of argine glycine aspartic acid. This particular ligand, RGD sequence, this actually helps in better cellular interactions and mineralization. And adding various materials to PCL, that is polycaprolactone, is not something new. There is a huge literature which talks where they have mixed ceramics, multi-wall carbon nanotubes, reduced graphic oxide, etc. with that of the PCL. In all these cases, there is an improved bioactivity, there is an improvement in the mechanical strength. At the same time, the most important point, note down here, is when PCL degrades, it creates an acidic pH environment during the biodegradation process. But none of these materials actually helps to neutralize this pH. There is a huge set of literature showing silk actually helps to neutralize the pH. So another set of literature showcasing the combination of silk with PCL, and two silk variants are being used, Bombyx mori and Antheraea mylitta. In both the cases, silk is used in the form of regenerated silk fibroin solution, and this solution is coated over the 3D printed scaffolds. So there's so many variables here: the coating time, dipping time, the concentration of the solution, and the penetration of the solution into the PCL scaffolds. And in addition to this, there is no single protocol where they can form regenerated silk fibrin solution from both the silk variants. For Bombyx mori, via a chemical treatment of lithium bromide is done. For Antheraea mylitta, it being a wild variant, it cannot be solubilized. So the direct silking from the gland of the silkworm is being done, which itself requires a lot of expertise. So in our study, we have developed the single protocol to form the silk fibron microfibers from both the cocoons. So for initially a process of degumming, then alkali hydrolysis was done, dialysis, and then lyophilization to form the silk fibron microfibers. As you can see, the same images of these silk fibroin microfibers, Bombyx mori and Antheraea mylitta. The length and width for Bombyx mori was around 18 to 18 microns and 5 microns respectively. For Antheraea mylitta, 21 microns and 7 microns respectively. These respective microfibers were mixed with polycaprolactone to form the composite via a process of wet mixing. And then we have optimized all the 3D printing protocols: the temperature, pressure, speed, the needle offset, the laydown pattern. All these parameters were optimized to successfully 3D print the PCL-silk scaffolds. Next, we have characterized these scaffolds. Field emission scanning electron microscopic images of these 3D printed scaffolds were done to observe the morphology of these scaffolds. As you can see that Bombyx mori with PCL have shown more roughness when compared to that of Antheraea mylitta with PCL. The reason for this is the density of Bombyx mori is less, so to form that particular percentages, we need to add more amount of Bombyx mori when compared to that of Antheraea mylitta. And with the addition of silk, there is an increase in the whole roughness of these scaffolds. Next, coming to the atomic fluorescent microscopic imaging. Here we have used a Gaussian software to do the analysis. Here also you can see that with the increase in the silk content, so the composites have shown more amount of roughness and more amount of root mean square values when compared to that of the pure PCL scaffold. Next, we have undergone compressive testing to understand the strength of these 3D printed scaffolds. PCL with Antheraea mylitta have shown the highest compressive modulus when compared to PCL Bombyx mori as well as pure PCL scaffolds. So with the addition of silk, we could say that there is an increase in the strength of these scaffolds. Next, to understand the printability, we have took the patient's CT data, which is in the form of a DICOM format, that is converted into an STL format, which is a 3D model. This 3D model is sliced down to 30% of its original size in X, Y, and Z direction to print the patient defect specific model. This patient defect specific model, which is the mandible, is being 3D printed with PCL with Antheraea mylitta composite. So, to summarize, we have obtained both the silk variants, Bombyx mori as well as Antheraea mylitta. We have formed one single protocol to form silk fibroin microfibers via a process of degumming and alkali hydrolysis. And then we have mixed them with PCL to form the composite, and we have optimized all the 3D printing parameters to successfully print the patient defect specific model. These are the references. I sincerely thank Dr. Falguni Pati, Associate Professor from the Department of Biomedical Engineering, IIT Hyderabad, and Dr. Mustafa and Professor Syed Masood from the Department of Mechanical and Product Design from Swinburne University for guiding me all through my journey, and special thanks to Composite Material Congress for giving me this platform. Thank you.